Protein and amino acid digestion and absorption. This slide presentation reviews the process of protein digestion and absorption, where protein structures are broken down to smaller peptides and individual amino acids. This process, like with carbohydrate and lipid digestion, involves the mouth, the stomach, and small intestine. Protein digestion is unique in that it requires the activation of a number of inactive zymogens, or pre-enzymes, to continue with the digestive process, particularly in the stomach and small intestine. Each protease enzyme is involved in digesting a different type of amino acid. You can see in this diagram the steps of the process of protein digestion in the stomach and small intestine. And these sites are particularly where the protease enzymes are involved with cleaving the peptide bonds. First, when protein-containing foods enter the stomach from the mouth, gastric cells release the hormone gastrin. So you can see the first step here. The hormone gastrin promotes the release of gastric juice such as HCl, or hydrochloric acid. This process denatures proteins, unfolding them to their primary structure, the amino acid chain. Also, you see here that hydrochloric acid converts the first inactive zymogen, which is called pepsinogen, to the active protease enzyme called pepsin. So this release of the gastric juice also is involved with activating pepsinogen to pepsin. Pepsin in the stomach then begins cleaving peptide bonds. The partially digested proteins then enter the small intestine, and this movement of the partially digested proteins from the stomach into the small intestine promotes the release of the hormones secretin and cholecystokinin. So that's the next step here. The partially digested proteins enter the small intestine, causing release of secretin and cholecystokinin. These hormones stimulate the pancreas to release additional inactive zymogens in the small intestine. And that's the next step here. These additional zymogens include trypsinogen, chymotrypsinogen, and procarboxypeptidases. Next, the inactive zymogens are activated, which further promotes the cleavage of peptide bonds and breaking the amino acid chain into smaller peptide units and free amino acids. You can see in this diagram an outline of each of the inactive zymogens, the signal activating each zymogen, and the site where they're active. So you can see here each of the inactive zymogens, the compound activating each. We see the active protease enzymes, the site of their activity, and the compounds they digest. And then here are going to be the end products of their digestion. So first we see pepsinogen, which we previously saw was released in response to increase in the hormone gastrin. Pepsinogen is converted to the active protease pepsin by HCl, or pepsin. It begins cleaving peptide bonds of amino acid chains in the stomach. Next, in the small intestine, we see activity of the remaining zymogens, trypsinogen, chymotrypsinogen, and the procarboxypeptidases. These again are released in response to increases in the hormones secretin and cholecystokinin. First, in the small intestine, we see trypsinogen, the zymogen, the inactive zymogen converted to trypsin by the activator enteropeptidase, or trypsin. Next, we have chymotrypsinogen activated by trypsin to chymotrypsin. Then we have the procarboxypeptidases activated by trypsin to either the carboxypeptidase A, carboxypeptidase B, or the aminopeptidases. So you can see the primary activator of these zymogens in the small intestine is trypsin. Once activated, each of these enzymes are active in cleaving peptide bonds in the small intestine. You can see while trypsin cleaves peptide bonds primarily of basic amino acids, chymotrypsin cleaves peptide bonds of aromatic amino acids, methionine, asparagine, and histidine. Carboxypeptidases cleave the peptide bonds of C-terminal either neutral amino acids, C-terminal basic amino acids, while the aminopeptidases cleaves peptide bonds of the N-terminal amino acids. The primary end products of protein digestion are primarily dipeptides and free amino acids. Next is the process of protein absorption, which, like with carbohydrate and lipid absorption, occurs in the villi and microvilli of the small intestine, where dipeptides and free amino acids are absorbed through the brush border membrane of the enterocytes. So you can see here the end products of digestion are transported from the lumen of the intestine into the body, and this goes first through the brush border membrane of the enterocyte and then the basolateral membrane of the enterocyte into the bloodstream. There are two mechanisms of protein and amino acid absorption. The first is the peptide absorption process, and next is the single amino acid transport process. There are peptide or amino acid carriers required for each of these mechanisms. Peptide mechanism of absorption is the primary means of absorption, the secondary being the single amino acid transport. So we first see the peptide transport, again the primary mode of entry into the enterocyte. As the primary mode, it occurs more quickly than single amino acid absorption, and you can see the diagram here of this process. So here we have the enterocyte, here's the lumen of the small intestine, and here's the bloodstream. We have the brush border membrane of the enterocyte, which is where we have our peptide carrier called PepT1, and then we have the basolateral membrane, which provides access to the bloodstream. You can see here the process of peptide absorption. So we have primarily our dipeptides entering through the PepT1 carrier through the brush border membrane of the small intestine. At this point, we have the dipeptides within the enterocyte, and they are hydrolyzed to the individual amino acid components. You can see there's also a series of ion transport processes occurring with the dipeptide process. So first, with the peptides entering the enterocyte, we also have a hydrogen ion that enters. But to prevent an accumulation of hydrogen ions circulating within the enterocyte, which would increase the acidity of the environment of the cell, there's a process pumping the hydrogen ions out, and then there's a sodium ion being pumped in, and then there's the sodium-potassium ATPase pumping the sodium out, and then the potassium back into the cell. The next process of absorption is the secondary mechanism of single amino acids. This involves specific carriers used to absorb single amino acids from the lumen of the small intestine through the brush border membrane of the enterocyte. Because each amino acid structure varies significantly based on the characteristics of its side chain, or R group, there are a variety of carriers used in this process, each specific to one or more amino acids. Most of the single amino acid carriers are sodium dependent. While this process is slower than the absorption of peptides, the essential amino acids, and particularly the branched chain amino acids, including leucine, isoleucine, and valine, are prioritized and are absorbed through this mechanism the most quickly. You can see in this diagram the process of single amino acid absorption. You can see how most of these carriers are sodium dependent, and you see here how the binding of sodium increases the carrier's affinity for each amino acid. So we see first the sodium binding, and then it increases the affinity of the amino acid, and then we have the amino acid being absorbed through the carrier into the enterocyte. There's also an ion exchange involved. So with the entry of the sodium into the cell, there's activation of the sodium-potassium ATPase, promoting the exit of sodium from the cell and the entry of potassium. Lastly, the amino acids now circulating in the enterocyte will need to be transported through the basolateral membrane into the bloodstream. This process is facilitated by single amino acid carriers and is similar to the process of the movement of single amino acids through the brush border membrane. However, many of the carriers for this process are sodium independent, so this process of transport through the basolateral membrane. There are certain genetic defects that affect the activity of some of these transporters. Particularly, the transporters used to absorb basic amino acids such as lysine and arginine, and these defects decrease the absorption of these amino acids. This leads to growth defects, increased circulation of ammonia, muscle weakness, among other symptoms. Another condition called Hartnup disease affects the absorption of tryptophan, which can lead to niacin deficiency. We will further discuss the relationship between tryptophan and niacin in future lectures.